Morning children, and welcome back. Last class we have started a new chapter, which is principles and process of biotechnology. Okay, anyone can say what is biotechnology? Yeah, it is the integration of natural science, organisms and molecular analogues for products and services. Okay, now what are these molecular analogues? Okay, tell me before that, what are the two steps in biotechnology or genetic engineering? First step is to alter the genotype or chemistry of the genome of an organism by introducing a gene from another organism, desirable gene from another organism. That is the first step. Okay, second step is, what is that? Create an ambience, create a situation, environment for it to get incorporated into the genome. That is the second step. Okay, now just now we told cutting a DNA from another organism and introducing into another organisms, right? Okay, now how is this cutting of DNA possible? With what do they cut these DNA? Yes, we are talking about the molecular analogues. Okay, now what is this molecular tools, which are the tools used in biotechnology or DNA technology? What is that? Which is called as I mentioned in the last class. Anyone remember? Yeah, it is the restriction enzymes or restriction endonucleases, whatever we can call them. Restriction enzymes, which are also called as molecular scissors. Okay, now see, this first step is the restriction enzymes, the molecular scissors is used to cut the DNA. Cut the DNA here. And after cutting, it is introduced into the plasmid. This is the plasmid. It's an artificially synthesized plasmid, children. We'll be learning it later. Okay, which is used as a vector. You know what is a vector? In transmitting the gene. This is the gene, cut gene, which is introduced into the plasmid. Okay, and then it is again transferred. This is a vector, children, vector, and it is used to transfer the gene into bacterial. Here it is used, bacteria is used as the organism for it to grow, right? Outside it cannot grow, you know that. Okay, so it is introduced into the bacterial cell, and here it will continue to replicate and form many copies, which is called as cloning. Okay, right. So what are these restriction enzymes? So they are actually enzymes or substances derived from nucleus of an organism. Nucleus, right? And so they are called as nucleases because they have been derived from the nucleus of the organism. They have been called as nucleases. And nucleases are of two types, which are endonuclease and exonuclease. What are they? Endonucleases and exonucleases. Okay, now what is this endonuclease? Endonuclease cut the DNA segment. See here. Cut the DNA segment at a specific point. What is the specific point? Specific point is that a palindrome site, it will recognize a palindromic site of 6 base pairs. You know what is a palindrome? Palindrome means if you read from left and right, you will be reading the same, right? So it will locate specific points of palindromic site of 6 base pairs. 6 base pairs. And cut it. They will not cut the DNA like this, children. They will not just cut it into two pieces, not like this. Right? They cut it in such a way that they leave like this. See, one is little longer, like this. Okay, they cut it in such a way that it will act like this. Here it will act. And it will cut in such a way that they leave sticky ends. Here are base pairs are there, base pairs are there here. Base pairs are there. Okay, now these base pairs are, they can attract any another purines or pyrimidines. So they are called as sticky ends. They are called as sticky ends. Right, now let us describe about the nucleases first, then we'll go into this in detail. Okay, children? Now, why they are called as restriction enzymes? In 1963, two enzymes which are obtained from nucleus of the organism, which were responsible for restricting the growth of bacteriophage. What was that? Restricting the growth of bacteriophage in E. coli were isolated. What did they do? They could, in a bacteriophage, these two enzymes could restrict, that is, stop the growth of the bacteriophage on E. coli. Okay, now these two enzymes, one of which cut the DNA, as I told you, cut the DNA, which is called as restriction endonuclease, and the other one added methyl groups to the DNA. One is adding methyl group to DNA, or it acts at the end of the nucleotide. That is why it is called as exonuclease. Exonucleases. The other is endonuclease. What do the endonuclease do and exonuclease do? The exonuclease act on the end of the DNA, which will add methyl groups to DNA. Okay, and the second one is will cut the DNA at specific points called as palindromic sites. Okay, now more than 900 restriction enzymes have been isolated from over 230 strains of bacteria. Now, coming to the naming of these restriction enzymes. Now, 900 enzymes have been isolated. How to name them? Yeah, the very first restriction enzyme obtained was HIND2. What is that? HIND, H-I-N-D, HIND2. Okay. Now, E. coli is another very famous restriction enzymes, right? Now, how is this naming done? E represents, so it is written in capital, right? Which is belongs to genus, naming of the organism, same like thing only. Genus name, E comes from E. coli. This E comes from the genus name. Co represent the species coli, okay, and Ri represent the strain, and R1 or R2 or numbers are given accordingly. Numbers are also given, Roman numbers. 1, you know how to write Roman numbers, right? Like that it is given, this R1, right? So Roman number, then order in which the enzymes were isolated from that strain of bacteria. What is this Roman number? Yes, the order in which the enzymes were isolated from that strain of bacteria. Okay, so as I told you, the exonuclease, they remove nucleotide, that is the act at the end of the nucleotide, children. Okay, they remove nucleotides from the ends of the DNA. And endonuclease, they cut a specific position within the DNA. Okay, see children, this is the sticky points I'm telling. These are the sticky ends. These are the sticky ends. Okay. See, when I told you, you're cutting a DNA from one organism and placing it in another organism, right? Now, the same kind of restriction enzyme should be used in both the organism, because then only it will cut at the same specific point. Okay, so this is the vector DNA. This represents this left side, which is written in red, which is done in red, is represents the vector DNA. That is into which the new recombinant DNA should be introduced. Okay, now this represents the foreign DNA from some other organism with the desirable genes is cut. Okay, and these both are cut at the same specific palindromic site. That is important, right? And they are combined, they are joined using, now I didn't tell you the name of the enzyme here. What is the name of the enzyme? That is ligases. To cut, we use nucleases. To stick, here we are sticking the two DNAs of different organisms, right? Which is done by using ligase. Okay. See, the first restriction endonuclease is HIND2, which whose recognition sites are this one. This is the recognition site of HIND2 and restriction enzyme endonuclease, right? See, this is what I told about palindromic site. GAATTC, and this way when you read again GAATTC, right? You can make yourself your own palindromic site of 6 base pairs. Your own you can make, right? And see, I told you about the sticky ends. These are the sticky ends, right? So restriction enzymes cut the strand a little away, not just, I told you they don't cut just exactly in the center into two pieces. They cut the strand a little away from the center of the palindromic site between the same two bases on the opposite strands. And this leaves a single-stranded overhanging stretches at the ends. They are called as the sticky ends. Okay, now the sticky ends are joined by DNA ligases. Sticky ends forms hydrogen bond with their complementary cut counterparts. This stickiness facilitates action of DNA ligase. Okay, right. The same thing is a schematic representation of the same. Whatever we have done the previous thing, same thing is done in a schematic way. Okay, should be drawing all these sketches from your text in your notebook. Clear, children? Okay. Thank you, all of you.